Good afternoon, everyone. I'd like to welcome you to the 2022 Burnet Institute Life Science Information Session for our Life Science discipline. Before we begin, I'd like to acknowledge that the traditional custodians of the land on which we meet today and pay my respects to the elders past and present. I extend my respect to any Aboriginal or Torres Strait Islanders who are present today. In Australia, Burnet Institute is located on the traditional land of the Boonwurrung people, and we offer our respects to their elders. We recognise and respect the continuation of cultural, spiritual and educational practices of Aboriginal and Torres Strait Islander people of this land. Here's our program for today. We have a range of our supervisors and researchers, including a couple of our students joining us as well. We also have, just to let you know, we do have a competition today. So if you do ask us a good question, feel free to ask us a question. Make sure you include your name. We have two $50 gift cards to be given out, and you must be on at the end to be in the running for these. So without further ado, I'd like to introduce our first speaker, Professor James Beeson.
Thanks very much for that, Brendan. So let me just show some slides here. Okay, well, welcome everyone. Thanks for joining us this afternoon. I'm just going to spend a few minutes, to say a few words about Burnet as a whole before it moves on to the program and have a range of speakers talking about particular topics and projects. Well, we're a medical research institute. We're independent, non-profit organisation, and we're very much solely focused really on achieving health impacts, improving health in Australia and internationally, particularly focused on underserved and underrepresented communities, those who need health improvements the most. It's just showing you our vision, which is a more equitable world through better health, and our purpose, that is what we're here to do, we talk and think about a lot, is create and translate knowledge into better health so no one is left behind. So that really tries to capture our thinking that we really want to improve health for everyone and give everyone the best opportunities for having better health and better well-being. One of the things that's a little different to Burnet, about Burnet to other organisations, is that we have this structure of our health programs listed there is the circles, and our disciplines, which are our areas of technical expertise and grouping people together with common research interests. So our health programs are really focused on achieving those health outcomes. So the three big programs are disease elimination, health security and pandemic preparedness, and maternal, child and adolescent health. In disease elimination, a large focus of our work is on infectious disease elimination, but it also includes harm reduction and healthy aging. So if you're a student at Burnet, you will be within one or more of the programs, depending on your project, and you'll have a primary discipline, life sciences, public health or international development. And having these two things interlinked, one that's focused really very much on the health issues and achieving those outcomes, and the other that's focused on the technical expertise and the people that we need to achieve those outcomes. Having those two things interlinked will really help us achieve much greater impact. And as a student, I think gives you a much better experience as well. We have core values, which we wanted to emphasise: respect, equality, inclusiveness and diversity. We're very proud of our culture of really inclusion, welcoming everyone and including everyone and giving everyone the same opportunities, creating a very positive environment for students and staff. As an organisation, we've got over 450 staff and students. We obviously based here with our head office in Melbourne, but we have activities throughout South Asia Pacific region, in East Africa and Southern Africa. We actually have offices in Papua New Guinea and Myanmar, coordinating and leading our work in those countries and also in the region. And we include a real spectrum of work. We've got our discovery research, population health, clinical trials, disease surveillance and prevention, and health system strengthening. So if you're working on a particular disease, you may be working on a particular element like life sciences, maybe it could be on immunity or pathogenesis or vaccines or drugs, but you really see the whole picture of the disease. And that's an important part of the way we work. And I think it's a really valuable experience for students. Then we have 3 integrated elements to the way we work and think about problems that we're working on. So one is, so that's the programs, the practice, the way we do our work, and the populations, the people that we work with. So firstly, our health programs, I mentioned these already: disease elimination, health security and pandemic preparedness, maternal, child and adolescent health. Secondly, then we have our domains of practice. So what's the technical expertise and who are the people skills and resources that we need to achieve those health outcomes? So that can be discovery and translation, data and modelling, interventions and implementation work, people partnerships and equity, systems and services, and then education and training. Particularly, thinking about students, we take honours, masters, PhD students, and have had many students over the years. And then lastly, the population. So we think about the health problem, but we're always very conscious of the populations and the communities that we work with or who we're aiming to benefit. And they're listed here, really a whole range of different populations, both in Australia and internationally, very much focused on achieving maximum health gains for people who need it, people who need the most, and working in that integrated way. And with projects that you're doing at Burnet, if it's in life sciences or in other areas of the institute, sometimes the outcomes are in the near term, sometimes they're medium term away, but some of our work has a longer term. So discovery science may have longer term benefits, but all the time, all of our staff and students are thinking about the problem and the ultimate way, the goal of impacting on the particular disease or health issue that we're working on. So I think that's enough from me. Thanks again for joining us, and hope you enjoy the session this afternoon, and encourage you, as Brendan has, to ask as many questions as you can and follow up with specific supervisors. Thanks.
Great. Thanks so much, James. That was a really great introduction to Burnet. Next up, we have a pre-recorded presentation from Professor Gilda Tachedjian.
Welcome everybody. I'm Professor Gilda Tachedjian and I head the Life Sciences discipline at the Burnet. And it's my pleasure to give you an introduction to the Life Sciences discipline. Burnet is a program-driven institute where our 3 programs capitalise on the broad technical breadth of our disciplines in life sciences, public health and international development to increase the impact of our work. All staff and students in life sciences are located on level 7 of the Alfred Centre, which is on the Alfred Hospital campus in the city of Melbourne. So it's a building on the left. We have amazing views of Melbourne CBD and Fawkner Park, which is just across the road. And on level 7, we have extensive office and laboratory space where we can do our research. In addition, we are only one of 2 institutes in Melbourne that has PC3 or physical containment level 3 laboratories, which are under negative pressure. And that allows us to work with infectious agents such as HIV and SARS-CoV-2. So here is our lab operations manager outside the PC3 and one of our researchers wearing appropriate PPE inside the PC3 lab. Currently, we have over 100 staff in life sciences discipline with 16 working group heads and around 28 students that come from different universities around Melbourne. And on this slide, I've included positions post-study completion secured by some of my honours and PhD students. So for honours students, these jobs include being a research assistant or working in a diagnostic lab or as a sales rep or in fact doing a PhD. And for those who've completed their PhD, they've either undertaken postdoctoral studies overseas or in Australia, or have secured a teaching position at a university, or a job in industry. Students at the Burnet have access to a variety of research platforms, including high-resolution microscopy on level 7 and through managed microimaging, flow cytometry, and protein production and robotics for high-throughput sample processing as part of our translational research facility. And at the Burnet, we have several flagship initiatives, and I wanted to specifically highlight the Burnet Diagnostics Initiative, which is on level 7, where they develop inexpensive point-of-care tests for a variety of diseases, including COVID. And we also have translational programs in developing mRNA-based vaccines for SARS-CoV-2 and malaria. Our purpose at Burnet is to create and translate knowledge into better health, so no one is left behind by solving global health problems in key populations. The pathogens and diseases we work on include viruses such as SARS-CoV-2 and HIV, vector-borne diseases with a particular focus on malaria, and we also work on aging and immune disorders. Our work focuses on solving fundamental questions in biology, as well as understanding the mechanisms of health and disease through analysis of samples from patient or populations. Our technical discipline strengths include infectious diseases, cell biology, protein function, immunology, including seroepidemiology studies, and we have growing expertise and interest in genomics, transcriptomics, metaproteomics, and culturomics. We have a laser-like focus to translate our findings into new drugs, vaccines, and diagnostics through collaboration with international development, public health researchers, and clinicians to make an impact on global health. Our life sciences working group heads are on this slide, and the names of researchers who have projects for next year are highlighted in red, which you can read in the booklet. And some of our researchers will be providing snapshots on their projects in this session. With that, thank you for your attention. And if you have any questions, please don't hesitate to contact us.
Thanks, Gilda. I'll video Gilda. Next up, we'll have one of our PhD students, Ellen Kearney. Thanks, Ellen.
Thank you. I will just share my slides. So thank you, everybody. I am a final year PhD student in the Malaria Infectious Disease Epidemiology group at the Burnet. And I also undertook my honours at Burnet in 2018. So if you've got any questions about honours as well, happy to answer them. So the research within our working group largely focuses on malaria. And we have a specific focus in the Asia Pacific region, largely in the Greater Mekong subregion. So the countries of the Greater Mekong subregion in Southeast Asia have made significant progress towards their agenda to eliminate all malaria from the region by 2030. They've significantly reduced the burden and associated mortality of malaria by 80% and 90% respectively, as shown in these GIFs here. However, as malaria transmission declines, the transmission of malaria becomes increasingly heterogeneous and localizes in discrete foci or hotspots, which are shown in these GIFs as well. This has important implications for the delivery of malaria control and elimination interventions, as a one-size-fits-all approach becomes increasingly inefficient. So the WHO have therefore made calls for a strong and sensitive surveillance system and identified this as a cornerstone of the elimination agenda, so that we can identify and target areas with the greatest need in order to ultimately accelerate progress towards elimination. However, as we move towards this elimination goal, the detection of the few positive cases of malaria becomes increasingly challenging. So when a Plasmodium infected Anopheles mosquito bites a person, which is shown in here, the parasite travels in mosquito saliva and it's injected into the human host, which causes the host to mount an antibody response. Serosurveillance involves detection of the antibodies against Plasmodium-specific antigens as biomarkers of exposure to malaria parasites, which allows us to detect people with a current malaria infection, as well as those who have had a recent malaria infection but have gone on to clear their parasites. We also use serosurveillance to detect antibodies against Anopheles salivary proteins, and these can be used as a biomarker of exposure to Anopheles bites. And so these serosurveillance approaches have been suggested as a useful tool with which we can monitor the complex and dynamic nature of malaria transmission, as well as to assess the effectiveness of vector control and malaria elimination interventions. So research within our working groups is largely based in malaria endemic populations, where we evaluate antibody responses to both parasite and vector biomarkers as a way to measure transmission of malaria and to evaluate the efficacy of interventions. We tend to combine laboratory-based research, so immunoassays and molecular techniques that we perform in pretty large cohort studies. So things are done in quite high throughput capacity. And then we combine these with quite advanced statistical and epidemiological analyses. And most research projects can kind of be tailored to suit the students' interests. So yeah, if you've got any questions, email me or Catherine or Freya, and we'll be happy to answer them. Thank you.
Great, thanks, Ellen. Next, we'll be hearing from Associate Professor Paul Gilson.
So I'm offering 3 projects, and one of these projects I'm going to be talking about today. So as Ellen just explained, there are hundreds of millions of malaria infections each year, resulting in more than 500,000 deaths. Resistance is spreading to all current drug treatments, and new drugs are needed. The current malaria vaccine is only about 30 or 40% effective in infants at preventing serious disease. And the current vaccine only targets the parasites that are introduced into the body through mosquito bite. And so we need extra protection with vaccines that target the disease-causing blood stage. And the proteins that the malaria parasites use to attach to red blood cells are currently favourite frontline targets for the blood stage of the parasite. So what I'm going to show here is parasites that are going to invade a red blood cell. So you can see the parasites here, these little things jiggling around. Red blood cells are the big circles, and there with the white arrow, a parasite just invaded a red blood cell. And you can see after several minutes that parasite will become highly amoeboid and then will eventually take over the entire red blood cell. As you can see it now converting into an amoeboid form. Now what we've done is we've tried to prevent these parasites from invading red blood cells by using antibodies to some parasite proteins. And normally these proteins would coat the parasites, preventing them from invading the red blood cells, but also recruiting immune factors that would come along and destroy the parasites. But with a recent set of antibodies we discovered something new. So here's the parasites coming out. They don't invade any of the red blood cells. They're kind of stuck on the outside. And then, hopefully, you'll begin to see that the parasites, we call these merozoites, start to become amoeboid. You'll see some arrows coming up. They start to grow these little arms and legs which come out. And this is what they would normally do once they had successfully invaded and were inside of a red cell. And so we're excited by this finding because it means we've somehow tricked these parasites into thinking that they can invade. And this may actually stop them from invading. And so that's really what the Honours Project is about studying this a little bit further. And so our hypothesis is that these invasion-blocking antibodies are neutralising parasites by tricking them into thinking that they've successfully invaded. And so that's sort of the main hypothesis of the Honours Project I'm offering. Thank you.
Great, thanks, Paul. Next, we have a video from Dr Josh Hayward, who will talk about some of the bat virus research on offer at Burnet.
Hi, I'm Josh Hayward, a senior research scientist here at the Burnet. Within the Burnet Institute's Retroviral Biology and Antivirals lab, we've been leading research into the relationship between bats and the viruses that they carry. Bats are exceptional animals, and one of their most infamous traits is their role as the hosts of numerous new and emerging viruses that are deadly when transmitted to humans, such as the Ebola virus and the SARS coronaviruses. A fascinating aspect of their relationship with these and other viruses is that bats don't tend to get sick from infections with viruses that are lethal to many other mammals. In recent years, we've begun to unravel the secrets of how bats manage to do this, and it's starting to look like it's a side effect, an eventual consequence of their evolutionary adaptation to flight, which is entirely unique among mammals. As the hosts of a great diversity of viruses, their antiviral defenses have been put under evolutionary pressure to adapt in ways not seen in other mammals, ways that we're just now starting to understand. Our bat virology and immunology research at the Retroviral Biology and Antivirals Lab, in collaboration with national and international partners such as the Australian Centre for Disease Preparedness at the CSIRO, have led to multiple breakthrough scientific findings over the last decade. We've discovered and described evolutionarily unique adaptations in the innate immune system of bats, among a special class of antiviral proteins called restriction factors. This revealed that their antiviral defenses have expanded and diversified in ways not seen in other mammals. By analysing the genomes of bats, we've also uncovered a huge diversity of genetic fossils called endogenous retroviruses, which are evidence of infectious retroviruses that infected bats across their evolutionary history. Now, more recently, our group, together with the CSIRO, reported the first ever discovery of an infectious bat retrovirus, which was found across a population of Australian fruit bats in New South Wales. Importantly, this virus is capable of infecting human cells, raising questions about the potential threat that it poses to people. Now, these discoveries have been published in prestigious and high-impact journals, and bat research is only one arm of research that we undertake in our research group. We also have exciting research underway in HIV drug development and research into the role of bacteria and their metabolites in the female reproductive tract and how they impact susceptibility to sexually transmitted viral disease. If you'd like to have a chat about possible projects, please feel free to get in touch with myself or the head of our working laboratory, Professor Gilda Tachedjian. Thank you.
Thanks, Josh. Next up, I'd like to introduce Dr Raffi Gugasyan, our Chair of Education at Burnet.
Thanks, Brendan, and appreciate the wonderful job you're doing for us as a student representative, as the student representative body. So thank you very much, Brendan. As Brendan said, I oversee education at the Burnet Institute, and one of the privileges that I have at the Burnet is to see outstanding young students pursuing their research careers and being able to assist them where possible. We take a lot of pride in this process, and I guess our mission statement is to ensure that students have the best possible research environment. Whatever they need, we'll endeavour to provide that for them. Research is a team activity for students. You won't be going through your research projects on your own. You'll be surrounded by, of course, your colleagues, fellow students, early career researchers, senior scientists, and group leaders. And of course, that's across a spectrum of research projects. We do have a diverse intellectual community, and we're very, very proud of being able to offer that to students. So cross-cutting research themes in projects such as healthy mothers, healthy babies, border security, disease elimination in hepatitis, HIV. So as you can appreciate, there's a diverse array of expertise there, and you'll have the opportunity to immerse yourselves in those areas. So we offer the opportunity to do field research, and that's particularly for our PhD students who may wish to experience being at the forefront of not only biomedical research, but also public health research. And that's unique to the Burnet. And in this case, we have Dr. Elise Wilson that you can see at Burnet's PNG Research Centre. So if you're interested in studying at the Burnet, how would you go about applying? And the first thing that I would say to all prospective students is to go to the link that I've shown here, burnet.edu.au@education. depending on what's of relevance for you, whether you're contemplating honours, masters or PhD, please do your research by accessing the 2023 student information booklet. You'll see a diverse array of projects there. Engage the prospective supervisors, email addresses are provided. Please send them your CV and academic transcripts if at all possible. Now, one of the key things is that you must also engage the respective universities within the country. And that's important, particularly if you're doing your PhD. In the case of scholarships, RTP scholarship applications are due at the end of October. So you will need to consider a project and then engage the supervisor and then apply for your scholarships through the university. So as I've said, enroll through the university. Also, please do consider tuition fees. It's imperative that this is considered, particularly for our international students who are contemplating coming across to Australia. Tuition fees play a significant part in your decision to come across. Upon acceptance, you can then register at the burnet and you'll be guided by your supervisor in terms of how best to do this. So here's the student information booklet. There's lots of interesting projects available. And as I said, please contact the respective supervisor associated. And we'll chat more shortly, particularly if you have any specific questions about studying at the burnet. I'll stop sharing and hand back to you, Brendan.
Great, thanks so much, Raffi. Next, we have another video from Callum Diamond-Smith and Josh Hayward again.
Hi, my name is Callum, and I'm an honours student at Burnet in the Retroviral Biology and Antivirals group. My project is titled The Interplay Between Human Endogenous Retroviruses, the Microbiome and Innate Immunity. Me and my supervisors are exploring segments of DNA which originate from viruses and how they interact with microbes and influence pathogen susceptibility in the vagina. This kind of research is really exciting because it hasn't been done in the context of the vagina. So I'm really excited to study something that's been uncharacterized in the field. As an honours student, my supervisors have really trained me to think critically when formulating hypotheses. It is so crucial to be able to look at data and not only be able to explain it, but actually scrutinize whether that is explaining it or it could be explained by other variables. As well, in my honours year, I've been required to analyse a lot more literature than I ever had to in the rest of my degree. And it's become so important to learn if a study is current, even if the data it presents is any good. My experience at Burnet so far has been really great. While the workload is definitely intense at times, I found the general culture at Burnet to be really relaxed. Everyone is extremely friendly and approachable. And especially for an honours student, people are really happy to help you if you have issues, even if they're not directly your supervisor. In terms of what this experience has given me in going forward into my career, I found it really helpful to see what an actual career in research looks like. My supervisors have really made me feel like a part of their team, so I'm not excluded from discussions about research other than my own, and I participate in lab meetings in which I get to observe their day-to-day at Burnet and what that actually looks like. As well, I can't understate the importance of me having these supervisors to give me invaluable advice about my career. They've all been where you've been, and they don't just oversee your research, but they really mentor you in becoming professional academics.
Hi, I'm Dr Josh Hayward, a student supervisor here at the Burnet. As a supervisor, it's really great watching a student grow over the course of the honours year. The honours year is often described as the most intense year of a research career, and part of the reason for this is the amount of change that a student goes through. There's so much to learn, so many new experiences to have, and of course, a fair amount of work. And this change as a student grows into a fully fledged researcher is just amazing to be part of. In terms of what we expect from a new student, I think that many students are surprised that we don't actually expect them to come to us with too many specific skills. We undertake biomedical research, so we expect a general but really only basic knowledge of the biological sciences, and we only expect minimal hands-on skill in the lab. This has always been true, but it's especially true for students whose undergraduate prac lab courses were affected by the COVID lockdowns. Anything that we need a student to do in the lab will be taught to them from scratch by our own expert scientists. More generally, we expect students to have, or at least to work on developing, good time management skills and good personal organisation. And there's very minimal hand-holding in honours, so we expect students to be on top of all of their coursework and deadlines. What we really like to see is students coming to us with a strong sense of curiosity and a willingness to learn a lot of new things. Our best students have excellent critical thinking and the ability to take what they've been taught and come up with their own ideas about their research project and what the findings mean. Students at the Burnet will be in a position to learn a lot of unique skills in the lab, and those skills will be specific not only to the particular area of research that they join, but also to the particular aims of their project. So there's a huge diversity in skills that students might learn, both inside and outside the lab. And what this means is that every lab-based research student in a given honours year will graduate with a distinct skill set that's likely to differ significantly from each of the other students that they graduate alongside. Prospective students can learn more about the specific skills that they'll learn by reading project descriptions and, more importantly, by speaking to potential supervisors. Now, after graduation, there are tons of different career pathways that can open up to new biomedical research scientists. I've seen students go directly to work in clinical diagnostic or R&D labs. I've seen others use their degrees as a springboard into fields such as medicine and business. And of course, many others continue on into higher degrees such as a research PhD so that they can further develop their abilities as independent research scientists and perhaps one day lead their own research in academia or industry. I highly recommend students check out the website, which has some of the potential projects listed. And if any of the research going on at the Burnet looks really interesting to them, don't hesitate to send an email to the head of the working group and line up a chat to talk about what projects might be possible, even ones that might not be listed. So yeah, get in touch.
Right. Thanks, Josh. I will now hand over to Raffi. Don't forget, we do actually have Josh and Callum live with us here as well as Tim, who's a PhD student also at Burnet. So yeah, feel free to pop your questions in the chat and I'll hand it over to you, Raffi, to lead us through a Q&A.
Thanks, Brendan. So we're basically opening the floor to questions and we can address those live by our panel experts, or we can respond in the chat. But let's go with live responses to begin with. Olivia, welcome. You've asked, hi, Brendan, why did you choose Burnet over other institutes? And what did you do after your honours? Did you go straight into a PhD? Brendan, would you like to answer that, please?
Sure, thanks for the question. I should clarify, I'm actually in the public health discipline, but the reason why I came to Burnet is essentially because of that. So we have public health, we have international development, and we have life science. And as it's already been pointed out by numerous presenters, Ellen in her presentation, that's a real good appeal of Burnet, that even if you're affiliated with public health, you know, you might work with life scientists or vice versa. And I've already on a couple of different projects, you know, where we use our cohort data and the life science people kind of analyze that for various different, you know, like one of the projects is markers of diabetes among people who use drugs, people who inject drugs. So definitely lots of different opportunities to go across disciplines. And between my honours and PhD, I worked as a research assistant through the Alfred Infectious Disease Department, which again works very, very close collaboration with Burnet Institute. So you don't necessarily have to go straight from honours to PhD. You know, it does depend on funding a little bit, but there's, I'll say more often than not, there might be an opportunity to work for a year or two as an RA while you're trying to decide what to do and get a bit more experience. Yeah.
Thanks, Brendan. Just elaborating a little bit on your research assistant position. So have you been doing that simultaneously, or was that before you started? Sorry, I didn't quite.
Sorry, so I started that 6 months after I finished honours.
Okay.
And did that, yeah, while actually also doing a master's, just kind of skip that little detail. But yeah, so started that after my honours,
Yes.
and I still do actually do 1 day a week research assistant work while doing my PhD as well. So that's another good opportunity, another good aspect of working at Burnet. My PhD is focused on hepatitis C among gay and bisexual men, but then I also work as a research assistant on hepatitis C projects among people who inject drugs to give you a bit more of a, broader experience. So that's definitely, sorry, shouldn't say definitely, but sometimes a possibility as well, just to give you a bit more, broader experience rather than just your own PhD project.
Right. Thank you. Greater diversity and support for you, which is important in this day and age, as we all know. I'll continue scrolling the chat. Thanks again, Brendan. Tommy has asked, what's the time frame that seroprevalence of malaria antigen is useful? That is, can you detect an infection that occurred 10 years ago? Perhaps I'll ask...
Ellen already answered that in the chat. Sorry, Raffi.
Have you? Thank you.
That's all right.
Much appreciated. Please do stop me
No worries.
if if it's already been addressed. And there is a question from Kai. I hope I pronounced that correctly. In the handbook, there's more than one supervisor. Do we email all of them, or would it be fine to email one of them? You can just email one of them, or if you prefer to increase your chances of getting that response promptly, just CC the other one in. That would be my recommendation for that, Kai. So thank you for your interest, and best of luck in moving forward. Moving along, we have a question from Emmanuel. I'm Emmanuel. I'm interested in malaria research at the James Beeson Laboratory. I'd like to pursue a PhD project discovering the mechanisms and targets of immunity against malaria. I want to ask if it's still available. I would initially say that if it's in the 2023 student information booklet, it's definitely available. If anyone would like to add to that and assist Emmanuel, please do.
I can add to that. So hi everyone, I'm Tim. I'm actually from James Beeson's lab. So everything on the, I'm not James, so I can't speak exactly for him, but everything on the project book anyway, it's because James has a lot of like ideas and grants. And so, yes, there is like a project, but you can see that it's quite vague. So obviously it's catered towards what we need in the lab, but also like towards like your like personal interests as well. So there's not like one specified project in that instance in James's lab. So I reckon have a meeting with him and see where that could lead.
Thanks, Tim. That's a great help. Much appreciated. Another question from Kai. Kai's a third-year microbiology student and wondering if you're still taking honours students. I believe that's for Professor Beeson, discovering the mechanisms and target of immunity against malaria as your EA. Ms. Collins told me to contact you at this session. Okay, so I'd suggest emailing Ms. Collins and she'll organise that for you promptly. I know that Professor Beeson is no longer with us in this forum. Moving along.
Sorry, Raffi, can I just jump in there?
Of course you can.
It's also a good idea to double check the university dates. That's probably actually, if anything, more important than Burnet to a degree, because they have very strict dates you need to submit your application, and I believe they do vary across the different universities.
Yes, that's correct. If you're referring to scholarships, absolutely.
Or even for honours applications.
Yes, yeah. For project cutoff for honours. Yeah, absolutely.
Raffi, I think that links in quite well with a question from Laura, which is
Please,
about...
I was just about to read it. So go ahead, Ellen. Yep.
Well, yeah, so I guess in my experience of that, it's the best kind of way to not work around it, but work within it is to contact the person that you're interested in doing honours directly, and they can normally sort it out through various affiliations. So I was a microbiology student for my honours, and my primary supervisor was through the Department of Epidemiology and Preventative Medicine. So we included a person who was Gilda, who was a member of Monash Microbiology, as a supervisor. So there is often a way to do it through the university department that you're interested in, but I think it's pretty specific in each case about where your supervisor has affiliations and who else you might be able to bring on board. So I think it would be kind of case by case basis, is my understanding, but Raffi would probably know more about that than I would.
Well said, Ellen. I think that's really helpful for students who are looking at studying at the Burnet. So every university and even sometimes departments have their own processes for enrollment. I think the expectation for most universities is that there is a co-supervisor from within the university department to connect with the Burnet supervisor. And they usually also have their own online application approach that students need to take. I think from the University of Melbourne, it's called, is it Sonia? I think it's called, yeah, the platform is called Sonia. So most cases, supervisors know about it. So I think the important thing is to engage the Burnet supervisor, and they'll get it firing back towards the university. And that process will work out quite comfortably, rest assured.
I guess I can add a little side point on that
Sure,
one as
Tim.
well, because for me, I didn't have like the highest GPA or WAM, and so I was a little bit worried about not being able to get a scholarship for a PhD. But this is kind of like the reason why I reached out to Burnet as well, because when I reached out to Professor Beeson, I knew that he had affiliation with both Monash and UniMelb, which means if I apply through him, I'm getting like a double whammy, like double the chance of getting a scholarship. Whereas if I apply for UniMelb only, if I didn't get that scholarship, then I'm kind of stuck. But also like just to speak to your potential supervisors, because even if you don't have the most competitive WAM, sometimes they have like affiliations, as Ellen already said, through other universities that you might or may not know of, and that can get you through the door into Burnet as well.
That's great advice, Tim. Thank you. I cannot emphasize enough for students to consider putting applications in through multiple universities. So, you know, in the state of Victoria, you might consider RMIT, La Trobe, Deakin, Uni Melbourne, Monash. You have lots of choice. Don't limit yourselves. The reason why I say that is because the cutoff scores are different for each university. And I think Tim has also emphasized that. So, you know, please do bear that in mind. I know it's duplication of your application, but that's also the easy part is that, you know, most applications require similar, very, very similar information. So I hope that helps. Are there any other questions that our attendees would like to ask? Please.
There's one in there, Raffi, about PhD enrollments only being at the start of the year, or whether there's an immediate intake.
Oh, thanks, Ellen. You know, the beauty of starting a PhD, you know, I love this part about it. You can pretty much start any time of the year. I think the key thing is in relation to scholarships, we have applications 4 times a year. 2 of those are domestic and 2 of those are international scholarship applications that the universities routinely accept. So you're governed a little bit by that, but you know, if you're coming with separate funding through other resources, for instance, NHMRC offer scholarships, then that creates greater flexibility in when you can start. But technically speaking, starting dates for PhDs are very flexible, in saying that common start dates for PhDs are at the beginning of the year, around February, March. But we do have a sprinkling of intakes throughout the year. Would anyone like to add to that?
I started my PhD in June. So
there you go.
Yeah, I finished honours in October, November, and then worked as an RA for 6 months. Took a 3-week holiday and then started my PhD.
Perfect. Just the refresher period, Ellen?
Yeah, it was good.
Yeah.
It's necessary. I'm not going to lie. It's good.
No, that's a great move, just to refresh a little bit. Honours is it is an intense year. It moves very quickly. So I think that's a great step to have a little break between honours and PhD. Ashley, I think there's a question from Ashley. Do you have recommendations for other organisations that give out a PhD scholarship that we can apply for, such as NH&MRC, like you just mentioned? I certainly do. Ashley, if I've understood you correctly, there are actually lots of funding bodies out there that offer PhD scholarships. And I know the challenge for prospective students is to identify those funding bodies that provide scholarships. So firstly, don't restrict yourselves to the RTP that the universities offer. There's a broad range of scholarships available. Consult your prospective supervisor. They'll have some good knowledge on other funding bodies that can provide you with a scholarship opportunity, at least to apply for one. Of course, you know, you can always look online, and they're not easy. They don't come up very easily, but our supervisors will have some knowledge on that. Would anyone else like to add to that? Tim, what did you do, by the way?
I would just like to preface this by saying that I was the winner of the gift cards last year, so keep the questions coming because it's not a scam. That started me off well.
I do recall that, Tim, actually.
Yeah. So I came from New Zealand. So at the time, I was still doing like all my research online for PhD supervisors, and I interviewed a few. I think that's a good idea, and also to ask general advice, to ask if you can talk to like maybe like a postdoc or like a student or even ex-students, because they would not hold back and would tell you the lowdown on what the lab is like. I guess I can talk a bit more about like burnet in general and what my experience is like if we have time and we don't have any burning questions.
Yeah, by all means, Brendan will let us know if we're going over time. He'll just chop us off, so it's all good.
Yeah, you can just cut me up anytime.
Yeah, no, go for it, Tim.
Yeah, so I mean, for me, I'm trying to give like a unbiased commentary. I feel like burnet has a more like professional vibe compared to like maybe like if you're at a university, which is it can be good or can be bad depending on what you're after, because everyone else here is like postgraduate and above. I feel like burnet is like very like mission driven, so like the projects themselves are more translational compared for life science anyway, compared to if you're in uni that could be more like discovery stuff. I feel like it's very collaborative, partly because everyone at the burnet, all the lab heads have adjunct appointments with different universities and institutions, so that lends itself very well to cross institutional collaborations. This is what I heard from, I didn't do my undergrad or honors in Australia, but everyone told me that compared to other universities or institutes, we have good facilities. I have a massive desk. I have 2 monitors and a laptop, courtesy of my lab. Yeah, I think some stuff that happens at burnet, even through lockdown, there was a mentoring program that really helped, especially when I'm coming from overseas. There's a whole other arm that Brendan already talked about the pop health and international policy arm. For me, my mentor was from that side. So I think it was good for me to know about what else is out there in my first year, and just talk about life in general. Our lab group is really tight knit. I feel like burnet really cares about like my personal well-being. We've had like student lunches and student symposiums, and then even more stuff to come out of lockdown, like end of year celebrations, and there's like a triathlon as well. So yeah, I'll put in a bit like downsides as well, potential things that you might not know. I have a scholarship, RTP scholarship from Monash. The hard thing to do is because most of those things are coming out of Clayton. I don't like to go in there for those student things. There isn't as big a student body compared to Monash here at Burnet, but as I said before, like there are still a lot of things happening. The good thing about that is that you can, if you see things that are happening close to you, because there's a MREP Monash campus here anyway, and you can still join stuff at Clayton. So it's like you can double dip if you want to. So it's not all bad. Yeah, and you can work as well, even though demonstrating in a lab at Monash will be hard if you don't want to commute to there. But as Brendan said, you can work as an RA there, like job opportunities at the offices here as well. So yeah.
Thanks, Tim. Great, great feedback and advice for our prospective students. We really appreciate that, and just emphasize that the students actually set the mentoring program up, and really proud of what they've done to get that going. Hats off to the students at the Burnet. It's a great program, particularly for those students who are just starting out, and we match them up with prospective mentees. And you know, again, credit to the students for doing that. Thanks again, Brendan. I'll hand back to you, please. Thanks everyone, appreciate it.
Yeah, great. Yeah, thanks, Raffi. It looks like the questions have kind of finished up. So thanks for answering those. But yeah, definitely feel free to email Paul, Josh, or any of the students on this if you have any questions. Yeah, we'll definitely be happy to answer. And if it's not directly related to our area, we can certainly work out where to direct you. But thank you very much to all of our speakers, our students, and everyone for joining. Have a great evening. And like I said, feel free to get in touch with us if you do have any questions.